Okay, I think we'll start. So, hi everyone, happy Tuesday. We're glad to present you another episode of Paleoperk seminar with our speaker Pedro Godoy. So, we have a tradition of having our outgoing members give a speech when they leave the committee. So, Pedro, thank you for your service to Paleoperks. And today, Pedro is going to talk about 200 million years of crocodylomorph evolution, systematics, paleoecology, and macroevolution. Before we get into the talk, for those who are new, we'll go over the format of today's seminar. First, we'll have welcome and announcements, which is ongoing, followed by the talk. Then we will have moderated question and answer session. And then Pedro has very kindly agreed to stay with us for tea time, which is an informal session. And ECRs are highly encouraged to join us. Don't forget to send your questions via chat to the questions at PaleoPerks host, who today is Rema. So a little bit of housekeeping announcements. PaleoPerks values the participation of all folks interested in the paleo sciences. Please remember to abide by our code of conduct during today's seminar. You shouldn't be able to unmute yourself, but in case you do, please mute yourself for the duration of the talk. You can ask questions by chatting to the questions at PaleoPerks host or by raising your hand. Uh, Using the raise hand function button. Technical issues should also go to questions at PaleoPerks host. We have closed captions built into Zoom. Use the CC button to show or hide them. PaleoPerks promotes the work of ECRs. Please nominate an outstanding ECR. The link to the form will be dropped into the chat window soon. Please also provide us feedback on our weekly feedback form. This is mostly to gauge the demographics of our audience. The form is anonymous and optional, but you're highly encouraged to fill it. Now, to introduce our today's speaker, Pedro Godoy. He has a Bachelor's of Science from the University of Sao Paulo in Brazil, a Master's of Science from the University of Sao Paulo in Brazil. He did his PhD at the University of Birmingham in the United Kingdom. He also did several postdocs, one in Stony Brook University in the United States of America, then in Universidade Federal. Okay, I'm sorry, I'm not able to get that name. And then currently he is an assistant professor at the zoology department of the University of Sao Paulo, Brazil. Pedro, please take on the stage and begin your talk.
Thank you very much, Aharna. Thank you very much for the introduction. Don't worry about the pronunciations of Brazilian universities.
I'm sorry.
It's fine. So let me just take over the screen share. Can you see it? All fine?
Yes.
Yeah, okay, cool. So yeah, thank you again, Harna. Thank you everyone from the PaleoPerks committee. It's a great pleasure to be here. I'm so excited to be on the other side of the stage this time. And well, and this is my talk. I mean, this is me. Thank you. I mean, Harna just introduced me, but before we go to the talk, I want to start with thanking PaleoPerks. Not only the people involved in the organization and I mean all the people that you already know, but also all of the speakers that made part of this, and of course all people that attend all of the seminars throughout these years. And from my side, I joined, I guess, in 2020. I'm never sure if it's 2020, 2021. I always joke about this. I think it was the end of 2020. I was host or co-host for more or less 30 times. It's not much considering how many seminars there were. So yeah, I could have done more of that. Participated on many different weekly meetings, edited many videos, which was cool. I used to like to do this. And of course, most importantly, I learned a lot from the committee, especially, but also from other people, from the talks, from other early career researchers, all the conversations we had, the special series that we had as well. So it was a lot, but it was very, 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 very important for me. So thank you very much. I want to thank, of course, all of the original committee, but also the new members that have joined after I joined, since I was the first one to join after the original committee. And yeah, all I can say is thank you. It was very important for me. It was very important for what ended up happening to my career in the end. So I learned a lot. Thank you very much. But yeah, let's continue and go to the talk itself. Again, thank you, Pedro Perks. So I guess, and I hope so, that most of you are familiar with the concepts of biodiversity. And of course, you can see here on the left an example of an environment with high diversity, like many different species. And on the other side here on the right, you can see a similar environment, but with lower diversity. In this case, biodiversity means biological diversity or number of species, basically. But there are other aspects of biodiversity or other concepts that also include this overall concept of biodiversity. For example, we can use different spatial references or different spatial scales to talk about alpha, beta, or gamma diversity, depending on the region we're talking about. Like alpha diversity is just like I showed you the example before, but beta diversity is when you compare the diversity of two different regions. And gamma is even a large geographical scale, or spatial scale. So it's those are some of the concepts we use to talk about diversity or to compare different regions. But there are many other examples that we can talk about when you're talking about diversity. For example, the genetic diversity, which is very important, for example, for adaptations. If a population has high genetic diversity, can more easily adapt to changes in the environment. So this is important for evolution, of course, and many people study this, quantify genetic diversity as well. Another example is functional diversity, which is more or less the role that different species play in the same ecosystem. So this is similar to ecological diversity, but a little bit different. And like food webs, or estimating or constructing food webs is a way of studying functional diversity, but there are many other ways of doing this. Another important concept that includes the idea of biodiversity is phylogenetic diversity, that depending on the type of data you're working on can mean different things. Here is the example that is mostly used for conservation biology, for example, which is basically the sum of the lengths of all branches in the tree. This is important to tell if a species is very different, phylogenetically different from the others that are closely related, for example. For for instance, for paleontological research, this is usually a bit different from measuring. This is a bit different, but yeah, it doesn't matter right now. Another thing is morphological diversity, also called morphological disparity, and I think this is more this is something that is more familiar to paleobiologists, for example. So it's basically comparing different morphologies from within the same group. We have examples here within insects or vertebrates and mammals, for example, which are great examples of a high morphological diversity or morphological disparity. And just to finish this short introduction, another example of diversity is what we call ecological diversity, that is can be understood as, for example, how a same group has displays different ecologies. And ecology here can mean different things. For example, diets, types of dietary strategies displayed by different species of the same group, or even different habitats that those species can occupy, again, talking about the same group, taxonomic group. Okay, so this was a very short introduction to show you different types of diversity, but now let's focus on the stars of this talk, which are the crocs. And all the living species of crocs or crocodilians are included in this clade called crocodilian. We're going to talk a little bit about different terminologies. And when I say crocs, what exactly I mean by this short term, popular term. But let's focus right now on the living species, the extant species, which are all included in the clade crocodilia. There are more or less 30 species today, and I say more or less because even though there are not many, we're not sure about how many exactly they are. Because as we are going to see, crocodilians or living crocodilians are very, very similar, and sometimes it's different to use morphology to tell the species apart. And there are recent studies suggesting that there might be cryptic sorry, species or hidden species that are revealed by genetic information or different populations that are genetically different, for example, but that we previously thought that were the same species. But doesn't matter right now. Just keep in mind that there are more or less 30 species. And they are divided in three families, which are gavialid, alligatorid, and crocodilid, as you can see here. They have some differences in terms of morphology between the species, but they look more or less the same as we have seen here in these drawings. They're more or less similar. They have, for example, most of them are large reptiles when you compare to all range of body size exhibited by other reptiles that inhabit the planet today. So their crocs are usually large. And they have similar body shapes, like elongated body shapes, usually elongated snouts. Even though there are snouts that are more or less elongated, they share similar morphological features. Similar with the teeth as well, position of the orbits and the nares, etc. This is how these three families are related to each other. And depending on the type of data you use, like molecular, morphological, and paleontological data, this can change a little bit, especially the position of this guy here, which is Tomistoma. It can be more related to gavialids or to crocodilids, but yeah, this is more or less how they're related. And this is their distribution in the planet. Here we have the whole countries painted to show that this country, there are crocodilian species, or almost the whole countries. But here in this, I prefer using these maps because they show more precisely the position or the distribution range of the species. Here for alligatorids and here for the other two groups. So this is how more or less they're distributed in the planet. And as you can see here and here as well, they are concentrated in the tropical regions of the planet. And this is very important for the message of the talk we're gonna see today. And so we said that there are where we can find them in the planet, and more or less like different species, different families, and more or less about their morphology, but also related to the morphology is their ecology. So because they are very similar morphologically, they are also very similar ecologically. So they're all semi-aquatic species, as you can see here. Of course, some species like the saltwater crocodile can go to the beach and enjoy a little bit. They're basically freshwater animals that some of them tolerate marine water, but they're mostly found in freshwater environments, semi-aquatic environments, basically. Some of them can walk and even run very well on the ground, but again, just look at them. They're semi-aquatic animals. They're fitted to stay in water most of the time. And related to that is their diet as well. So most of them feed on fish, as you can see, like longer snouts like these are usually related to a piscivore diet. But crocs can eat everything they found, everything that fits in their mouth. And if they're larger, like these famous Nile crocodiles, they can ambush a larger prey, and they're very good at doing this as well. So basically, they're carnivores, usually top predators of their environment. Okay, so everything that we have seen right now, and if you remember the introduction of the talk, talking about different types of diversity, I think we can agree that crocs have a low diversity, low number of species, low morphological diversity, and low ecological diversity as well, and also restricted distribution in the planet. So do we agree with this? But it's hard to say that they have a low diversity when we don't compare to other groups. And to make a fair comparison, we need to compare crocodilians with groups of animals that have a similar or somewhat similar evolutionary history. So let's start with the sister group of crocodilians, like birds, and even closely related reptiles such as turtles and even lepidosaurians or squamates and tuatara. So when we start with birds, for example, just focusing on the number of species right now, it's I mean it's it's not looking great for crocs. It's way more species today than for crocs, like 10,000 species for birds. And for lepidosaurians, like squamates and tuatara, also about 10,000 species. Again, yeah, way more than the 30 species of crocodilians today. Even for turtles like testudines, we have about 10 times more species today than we have for crocs. Again, turtles are not very famous for their high diversity, but still more diverse than crocs. And even in terms of ecology, we have marine turtles that we don't have something like this for crocs. We have completely terrestrial turtles. We don't have something like this for crocs either. And if we expand our comparisons to tetrapods, we also see similar scenarios. Like, for example, amphibians have about 8,000 species today, and mammals have about 5,000 species today. And particularly for mammals, many more morphotypes and ecological habitats. So it's, again, not looking great for crocs. So here in this graph, you can see the whole diversity of vertebrates. And here on this side, on this left side are tetrapods, and on the right side are fishes. And well, we can all agree now that crocs have a low diversity. If we compare the number of species of most of these most important groups of vertebrates, crocs are among the lowest numbers. We have some groups that have even lower diversity, just like tuatara and concephalians, the lungfishes or coelacanth and other stuff. But crocs or crocodilians are among the lowest diverse vertebrate groups. Okay, but it has not always been like this, and here is when we talk a little bit about the terminologies we're going to be using in the talk. So this clade here is called Crocodylomorpha, which includes crocodilians, that includes the living species today, but also includes loads of other species that are already extinct, but closely related to living crocs. So when I use the term crocs throughout the talk, I'm usually referring to this group, Crocodylomorpha, to the whole group. I'll try to use Crocodylomorpha to not confuse you, but sometimes I'll say crocs, and I'm usually referring to the whole group, Crocodylomorpha. And this group has about 500 species described. As I said, most of them, except for the 30 living species, are already extinct. And not only in terms of number of species, but also in terms of morphologies, they were very different. We're going to see this in detail throughout the talk, but I mean, there are some very fully terrestrial ones, fully marine ones, again, terrestrial crocs, some very, very big ones, even larger than the one we see today. Again, completely marine crocodiles, crocs, until we reach the living diversity today. So, and here just a little bit more about the diversity of forms you can see among Crocodylomorpha, among all this long evolutionary history. But again, we're going to see this in detail throughout the talk. So this is how the talk is going to work. And this is the main message I want to transmit to you. Because of all of this and because of all I'm going to say from now on, I do think Crocodylomorpha is an excellent system for evolutionary studies. And I'll try to show this to you during the talk, but I'll divide the talk in two main parts. So the first one, We're going to take a look at systematics and taxonomy, looking through the different groups of Crocodylomorpha. And then we're going to take a look at macroevolutionary patterns to really show you why this group is an excellent study system for evolution. And I'll do this showing some of my own contributions with colleagues, of course, to show some of the stuff I've been doing for the last years. Before that, let's a little bit of context, evolutionary context for Crocodylomorpha. I showed you that they are related to living crocodilians, but where exactly are they placed in the evolutionary tree? So they're members of a clade called Archosauria, which has two main lineages. One is AV Metatarsalia, which includes birds here in the top, and also non-avian dinosaurs and pterosaurs. And the other one is Pseudotsuchia, which includes crocs here, or crocodilians here on the top, but also Crocodylomorpha here, and other clades that we're going to see. Pseudotsuchia and as well wave Metatarsalia are originated in the end of Permian, so before the Permian mass extinction event, and they survived this mass extinction event. And a little bit after that, in the late Triassic, we have the first records of Crocodylomorpha. And this is the phylogenetic definition of Crocodylomorpha, but it's basically nearly all pseudosuchians, except for a few Triassic clades, such as Rhyzosuchiids, phytosaurs. Sometimes phytosaurs are on the side of the tree, but not always, and ethosaurs as well, and some other groups that are not as diverse as Crocodylomorpha. And by the end of the Cretaceous, Crocodylomorpha is the only Pseudotsuchia lineage that survives until today. So yeah, Crocodylomorpha is basically the crocodilian total group. That's another way of saying it. So let's go to the first part of the talk, in which you're going to talk a little bit about different groups of Crocodylomorpha. Again, taking a look at some of our contributions to this field. And I'm going to be using this figure a few times to situate you both phylogenetically and also temporally. So starting with this end Triassic temporal, forgot the strap, no, forgot the name, but this period of time. And so here we're talking about some of the first records of crocodylomorphs, and some of we don't have a monophyletic group that contains all of them, so they're usually called non-crocodiliforms crocodylomorphs, and are these guys here in dark green, basically. And this is very interesting because we can see that some of the first crocs were very, very different from the crocs we have today. And maybe the most striking feature is that almost all of them were very small and much smaller than the large species that we have today. But they're also fully terrestrial animals, and they were also gracile, and their feeding behavior was associated with an insectivore diet. So they were very, very different from the crocodilians we have today. This is very cool. Moving forward on this figure, we get to the Jurassic period, which has witnessed the origination of some very important groups we can see here. But maybe the most important diversification during the Jurassic was for this group here in red, called Thalattosuchia. And as the name suggests, Thalattosuchia were marine animals. Not all of them were fully marine. Some of them were possibly semi-aquatic, but related somehow to the marine environment. And even though there are some species that could be freshwater animals as well, but some of these guys had very extreme adaptations to the marine environments, to even a pelagic lifestyle, as you can see here. So just zooming in one of these specimens, as you can see here, this specimen had some adaptations that are somehow related to cetaceans today, and even ichthyosaurs in the past. So they had flippers, they had reduced limb size, they had the modification in the tail as well. So they were very, very adapted to the marine lifestyle. And related to this group, we have a collaboration to the knowledge of the systematics and diversity of this group, which was a paper published 2 years ago, which focused on a specimen found in the Jurassic coast of the UK that we named Turnisuchus. There wasn't much of this specimen, so these are the cranial elements we had. There were also some postcranial bones, and we CT scanned it. But the cool thing about this guy is that he was, by the time we described it, he was the oldest known Teledisuchian, and also because of the phylogenetic analysis we did, it was the basal-most. So it was situated as a sister group to the two main lineages, or to all other Teledisuchians, basically. So because of this, we also did some divergence time analysis to take a look a little bit at the origin of the group. And we do expect to find some Teledisuchians closer to the Triassic. So hopefully you're going to find some older specimens to elucidate a little bit more about the evolution of this group, which is very cool. And this is the paleo art done by Julia de Oliveira, Brazilian paleo artist, which is very, very cool. So it was not exactly very, it did not have those extreme adaptations to the marine environment yet, but it was, of course, a marine animal that was found in a marine environment as well. So moving forward to the Cretaceous, again, we see the diversification of some very important groups, including this blue one here, which is Neosuchia. But we're going to focus on these orange ones here called Nodosuchia. And Nodosuchia is a very interesting group because, I mean, it's my preferred group. It's the one I like the most among crocs. And it's for some reasons. Firstly, because they're found here in Brazil, but also in South America and in Gondwana in general. So they have a Gondwanan distribution. There are some specimens and species also found in the northern hemisphere, but they're diversified mostly in the southern hemisphere. And this is already cool. But they are also cool because they were very diverse. As you can see here, representing most of the diversity of the group, have nearly 50 species. And we consider that this is an completely extinct group, and that not all species are preserved in the fossil record. You can only imagine how large or how high the diversity of this group was in the past. And there were some, and maybe the most important feature about these guys is that there were some species that were very, very, very different from living croc that we see today. So there were some with like duck-shaped snout, some that were feeding on insects, some with very different teeth morphology. Like some of them have something that we call heterodonty, so teeth that were different along the tooth row, just like us mammals have. So this is very different when you compare it to a living croc. So that's why they were so cool. Most of them are fully terrestrial as well. And there are some of them that were very, very cool, such as Sinosuchus, that was in the cover of my talk, that based on the morphology, we can almost surely assume that they were herbivores. Mostly because of the morphology of the teeth, which are similar to what we see today here in marine iguanas that are herbivores. So yeah, the teeth of this guy were very, very bizarre. But look at the skull itself; it doesn't look like a croc, right? So yeah, very different from living crocodilians. Just a little bit more about this guy because he's very cool. But there were some Nodosuchians that were even so we have just seen a herbivore, we have seen some insectivores as well, but there were some Nodosuchians that were fully carnivores, even included in a category that we call hypercarnivore, feeding almost exclusively on flesh of other vertebrates, like Barysuchids, as you can see right here in the tree. And they were usually larger than most Nodosuchians, like about 3 to 4 meters long, and again fully terrestrial, possibly running fastly throughout the ground because of their postcranial morphology. And also based on their skull morphology, you can see some adaptations to this diet, like the reduced number of teeth, very large uh, uh, caniniform teeth as well. And we also can also mention some of the contributions we had to the knowledge of this group, the Barosuchids. This was actually my first paper publishing 10 years ago already. Well, that's a lot. That's a long time ago. In which we describe a new genus and species of Barosuchid. And this is the skull, and this is the remaining of the skeleton, what we call the postcranium. And in this region here, between the the ribs, we can see something very unusual. So focusing on this region here, zooming in, we can see basically this. So in green here, we can see the ribs. In orange, we can see the abdominal ribs, which we call gastralia. But in yellow, we see something very different. We see both cranial bones and teeth, which indicates and very much smaller than from the actual skull of the specimen, and also we have different morphology, especially based on the teeth. So based on this, we can see that, and based also on the relative position to the ribs here and abdominal ribs, we can say that these bones here were actually from the last meal of this Barosuchid. So this is a little bit of the field when we still didn't know that there were some very cool things going on in the abdominal region of this specimen. We collected it, took to the lab and prepared, and we found those nice bones within the belly of the large croc. And this is a representation of what could be going on. Based on the morphology of the cranial bones in the belly and also the teeth, we can be almost sure about the group that the prey belonged to, which was another Nodosuchian, another crocodylomorph that inhabited the same environment, and it was possibly herbivore or at least omnivore and smaller size as well. So it was very likely that Barosuchids were preying on them. So, but was the only so far direct evidence of this interaction. So moving on, and lastly, for this first part of the talk, we get in the Cenozoic here in orange, and we can focus on especially on this group here in light green, which is Crocodilia, which includes, as we have seen, the living crocs that we have talked about already. But besides the living crocs that are seen here in the rightmost position of the tree, we also have all these extinct diversity right here, which is much higher than living crocs today as well. So among these extinct diverse, we had, again, marine crocodilians and also more terrestrial crocodilians compared to the living crocs. And also in terms of morphology, we had some very different or some significant disparity between these extinct crocodilians. But I think one of the most interesting features or morphological differences that we see among these extinct species is in relation to their body size. So on one side, we had very, very small crocodilians like this one. It's hard to see here, but I guess this bar has about 10 centimeters, maybe something like this. I'm not sure, but this specimen was about 50, maybe less centimeters, maybe 30 centimeters. And on the other side, we had something very, very large like this, like the Purosaurus. This is just a skull. This guy had about 10 meters or maybe even more than that. And it's nice and it's funny to realize that both of these species, one of the smallest crocodylomorphs of all times and perhaps the largest one, belong to the same subfamily, which is called Caimanini, which includes the living caimans today here from South America. So yeah, and this subfamily in particular is very interesting, and I really like them, not only because we have lots of them in Brazil because they look very cute as well, but mainly because of this morphological variation we have within this very small group. It's not a very diverse group. It's a small group compared to the whole Crocodylomorpha. Here is a little bit more of this morphological diversity. So you can see here the differences in size between these species. And again, we have another contribution to the diversity of Crocodylomorpha. In this case, Camanini. It's not a new species. It was actually a redescription of a known specimen that was already described. And it was very nice because this specimen was collected in Argentina in the 1930s by the great paleontologist J.J. Simpson. It was collected and described by him. And I really like J.J. Simpson because of his works on macroevolution that we're going to talk about in the second part of the talk. And in this case, we were able to CT scan the specimen, to redescribe it, to redo a phylogenetic analysis, etc. It was nice that the specimen and the paper itself was highlighted in the cover of the journal. And here's just a little bit more about the morphology of the specimen. It's not much again. It's basically cranial and mandibular elements, not perfectly preserved, but with the CT scan data, we could look at the morphology a little bit further than before. Just a little bit of the phylogenetic results and time divergence time estimation again. Okay, so with this, we finish this first part of the talk, and we can move forward to the second part, which focuses on the macroevolutionary patterns of the group of Crocodylomorpha. And of course, macroevolution can be studied through different approaches. And I usually tend to think that there are two main ways of studying macroevolution, which are studying morphological evolution or phenotypic evolution, or diversification, which is like diversification rates or number of species through time, etc. And this talk, we're going to focus mostly on phenotypic evolution or morphological evolution, as you can see here, both because it's the aspect of macroevolution that I like the most, but also because of what was available in terms of data at the time we started doing this kind of analysis. And as you are going to see during the following slides, most of the results we have for this kind of study, morphological evolution or macroevolution for crocodylomorphs, is going to be focused on the cranial morphology of these animals, both because of historical aspects of crocodylomorph paleontology, but also a little bit of researchers' biases, because we're used to think that cranial morphology of crocs are more informative or more diagnostic, so we tend to describe or focus more on descriptions of the skulls than the other elements of the skeleton. And looking only at the living diversity of crocodilians, we can see that there is some variation both within the skulls and the lower jaws, but it's not that much. So here in this first example or paper I'm showing you, I use geometric morphometrics to assess cranial shape variation within the whole group, Crocodylomorpha. And as you can see here, there was way more variation when we consider the extinct diversity of crocodylomorphs than you can see in living crocodilians today. So here in dark green, we can see which each one of these points represent one different species. And here in dark green, you can see crocodilians, which includes the living species, but it's not restricted to them. So here, crocodilia here also includes the extinct diversity of crocodilia. But you can see here that a significant part of the morphospace is occupied by completely extinct groups, such as Nodosuchia here in yellow, and also Thalattosaucia here in dark red. And with this, we were able to play around with the data and get some nice results. For example, disparity or the variation of cranial shape among these groups. You can see that Nodosuchia has the highest variation. And you can see only here in this PC1 and PC2 plot, we can see that this is true. And Notosuchians again were those mostly Gondwanan species that included some of those weird guys that were herbivores, some of those hypercarnivores as well, some of those that had heterodonty. So it really makes sense to see those guys here on the top. And consequently, in terms of which type of ecology or ecological category displays the highest diversity or disparity or variation, we can see here that terrestrial crocodylomorphs had the higher disparity as well, mostly again related to the diversity of notosuchians that we have just seen. Still talking about cranial shape, there's another study that I want to show you, which uses the same methodology, geometric morphometrics, to investigate a specific case, which is very nice. So we had access to this skull of a juvenile barosuchid, which is that group with hypercarnivore specimens. And we noted that it's a juvenile, it's not fully adult, but it's not newborn. It's like it was possibly approaching mature stage, but it still clearly has some features that we can use to differ if it's from an adult specimen. And it was very nice because we noted that this juvenile skull of a barosuchid looked similar to adult skulls of other species of notosuchians outside of Barosuchidae. And this suggested to us that a very interesting evolutionary process could be taking place here, which is called heterochrony. And to investigate this, we again use a geometric morphometrics, cranial shape, to compare the shapes of skulls of different species of notosuchians, both within Barosuchid here in dark blue and without outside Barosuchid here in other colors. And here's the juvenile, here's the adult of the Barosuchid. So they are different. And by doing this analysis, we could see that the juvenile of Barosuchid, the skull of juvenile, is much more similar to adults of other notosuchians than to the actual adults of the same group. And because of that, we suggest that there is heterochronic process going on, which we classified as paramorphosis because we could not go further. There are different types of paramorphosis. We were not sure exactly what kind of paramorphosis was going on, but we're sure about the presence of paramorphosis, which is basically like the changes of features throughout the evolutionary, the ontogenetic development of the individual, and changing from features that in the juvenile already look like adults of other groups, but in the adult change it or develop further, usually related to something like, in this case, I think related to the extreme feeding habits of these guys. So reduced number of teeth, very tall skulls. So it's basically the exaggeration of these features that were present already in adults of other species that became even more dramatic in the adults of Barosuchids. And moving forward, so we're approaching the end of the talk, I want to talk mostly about one specific study, which is going to be focusing on the body size evolution of crocodylomorphs. And before we focus on crocs specifically, let's talk a little bit about body size. So body size is a very important feature for all animals. If we think about it, like body size is related to of course, the morphology, the whole morphology of the body of the individual, but also physiological aspects, also, of course, related to the evolutionary history of the species. So there's a lot of biological features that are related to body size that can somehow summarize all of these things in just one feature. And this is very useful, and that's why many studies or evolutionary studies have used body size to somehow investigate, quantify, characterize patterns, investigate processes, and etc. Specifically in paleontology, that's also true because even though we know that vertebrates, for example, are very rarely fully preserved, or the whole body is very rarely preserved. We can still use parts of the skeleton to estimate the total body size of species. And because of that, there's a lot of paleontological studies that focus on body size evolution of extinct groups. In the case of crocodylomorphs, we have this important contribution to the study of body size evolution of the group. It's not the only one. Mostly after that, there were some other studies that did something similar, but focusing on more specific aspects of body size evolution in the group. They're very nice studies as well. But in this case, since it was the first one, it was we tried to explore many different aspects of body size evolution in the group, and I'm going to focus on some of this. Just to make sure it's clear here, we use a cranial measurement to represent the whole body size of the individuals or the species, actually. Of course, again, because we didn't have the whole bodies or the whole skeleton preserved for all species, and the idea here is to maximize sampling. And here in this plot, you can see perhaps the most important result of this study, which is the result of the model fitting analysis. So we basically use the evolutionary tree of crocodylomorphs and the body size data to try to tell which evolutionary process was going on to generate this observed body size pattern. And we fitted different evolutionary models representing these different processes to see each one of these best fitted the data and the tree, of course. And among these, we have here below a simple Brown emotion, which is basically random evolution, a simple OU model, which is a more constrained mode of evolution, the early burst, which represents a very rapid initial diversification, or in this case, body size evolution at the beginning of close to the origin of the group, and some trend models that would represent a trend towards larger size or smaller size through time. And as you can see here, the best model was this model here in red, which is a surface model, which is basically a OU model with multiple regimes, or a non-uniform OU model. But what is a non-uniform OU model, or OU model with multiple regimes? Maybe the best way to represent what a non-uniform OU model would be is the idea of adaptive landscape, or in other words, or the other way around. The OU model with multiple regimes might be the best model to represent the idea of an adaptive landscape. And what is the adaptive landscape? Is this idea that both the environment and the species change, as we know, of course, through time. And these peaks here represent optimum values of traits that are, of course, if they are optimal, they're better adapted to the environment, or every selective processes that can be happening and pressures that can be taking place. And with time, of course, this adaptive landscape changes, and the optimum values of these traits can also change. And this idea is represented in this OU model. So we're going to see different optimum values for different situations or different parts of the tree, different time periods, etc. This is the way of representing this model for Crocodylomorpha. And each one of these different colors represent these different macroevolutionary regimes, and each one of them related to different body size values, or optimal values. And we can see here in dark green, this is related to lower optimum body size values, and darker brown here represent larger body size optimal values. And this is somehow related to, we have just seen the first part of the talk, in which you have seen the different diversity through time, or different groups through time in the crocodylomorph tree. So we know now that the first, or the oldest crocodylomorphs, the non-crocodiliform crocodylomorphs, are very small, and that's why they're associated with this dark green color, or lower body size macroevolutionary regime. And as we move, like Thalassocus have larger sizes in general, also related to larger body size macroevolutionary regimes. Notosuchians have a high diversity, that's why I have different colors representing them here. And we have some very large animals, as you can see some of these darker brown here, but not large groups, just some few lineages. And finally, until we reach the living diversity today, as you can see here on the right, which is basically dominated by large size, not largest as large as possible, but you can say they're overall large macroevolutionary regimes here. And so the second question we asked after we found out about the evolutionary mode that contributed to the observed pattern of body sizes or the distribution of body sizes. So the second question we asked was what could be driving crocodylomorph body size evolution? So there were some possible explanations for this. And maybe the one that comes to mind first is the environment, or in this case, the global temperature. So we compared the global temperature through time to the whole group, the body size of the whole group Crocodylomorpha, and also of different subgroups, like some of those who have seen the first part of the talk, like Thalassasuchians, Notosuchians, Crocodilia, etc. And overall, what I can say is that we did not find significant results or significant and strong correlation between temperature and body size, which means that overall body size is not being driven by global temperature.
You have 3 minutes left.
Oh, sorry. It's gonna take a little bit more. Please. Yeah, I'll try to rush a little bit. So yeah, overall, we didn't find any significant correlation between these two factors, but there was one exception that I mentioned very soon. So what else could be driving crocodylomorph body size evolution? So the second thing we looked at was the ecology of the species. And what it can, we can subdivide the ecology of, or the species of crocodylomorph in three main ecological categories, like as we have seen in the first part of the talk, like marine habitats, freshwater, and terrestrial. And we do find a trend, a pattern very clear here, in which you can see that marine crocs are usually larger than semi-aquatic, that in parts are also larger than terrestrial crocs. So you can see the distribution very clearly when we look at these two plots. So another paper went further to explore this relationship between ecology and body size of Crocodylomorpha. And this, I did not participate on this, so I'm just mentioning. And they find that this could be correlated to thermoregulation in water, which is different from thermoregulation in air. So they lose temperature more quickly in water, like any vertebrate. So that's why maybe crocs needed larger size when they transition to a marine habitat, and also because of lung capacity. So if the animal is larger, they can stay longer below water. So coming back to the results between temperature and body size, I said that there was one exception, and it was when we were looking exclusively to this clade here, Crocodilia, which includes the living taxa. So in this case, as you can see here, there is a correlation. So in red here, we have global temperature throughout the Cenozoic. And in green, you have mean body size values for crocodilian species through time as well. So you can see that as temperature went down, and we are currently, or until very recently, going through a global cooling event, the mean body size of crocodilians went up. So in average, they became larger. And we can try to explain this by looking at this plot fraction. For example, we can see that most crocodilians are large in size, but there are some smaller species, and there were some smaller species in the past, or lineages. And with time, they became extinct, mostly when the planet started to become cooler. But not only the very small ones, but also some medium-sized ones. That's why the average body size became larger through time. And this plot here maybe can explain what was going on. So putting everything together, body size, temperature, and also here you can see latitudinal distribution, so distribution in the planet. So here you can see the latitudinal distribution of species through time. So here closer to the present, here older species. The circle sizes indicate body size, but most importantly, species that are located here on the top are were found closer to the poles, and species closer to the bottom were found closer to the equator. So as you can see here, through time, we have a more restricted distribution of species until we get to the pattern today, in which we have seen in the beginning of the talk, in which the species are restricted to the tropical regions of the planet. But it was not always like this, mostly because of the planets that became cooler through time. So at the beginning of the Cenozoic, during the Paleocene and Eocene, they were able to inhabit regions that were closer to the poles because the planet was warmer, and as ectotherm animals, these species probably had to live in places with not as much temperature variation throughout the year or seasonal variation. And this changed with the global cooling event, with more regions of the planet becoming too cold for crocs, basically. And as you can see here, we have the current distribution, and the final message is that, like, with time, less space was available for crocs to inhabit in the planet, and maybe with less space, we started to see more things like this, in which the distribution of species were occurring in the same regions. So species were co-inhabiting the same regions. And because of that, maybe the larger species outcompeted the smaller ones, and that's why the smaller lineages became extinct through time as well. So this is just a hypothesis, was not tested, but it's just a final thing I wanted to mention to you. Sorry for me taking longer, and thank you again.
Nice. Thank you so much. For such a great talk. There were so many cool crocodiles.
Yeah, that's the best part.
So because we're nearly at the top of the hour, what we're going to do is that's okay. We're gonna go straight into tea time. So I'm gonna take a screen share over from you. So we encourage anyone that had a question to stick around for tea time, so you can talk to Pedro and ask him all your cool questions. But thank you very much to everyone for joining us. Please remember to fill out the weekly feedback form, and the link will be in the chat very shortly, so that we can learn a bit more about who attended today's seminar. And next week, on September 3rd, we'll be joined by Imran Ali from Petrostrat in the UK. And Imran will be talking about biostratigraphy and paleo careers. More along our paleo pathways style seminars that we've had in the past. So we hope to see you then, if you're not sticking around for tea time. If you are, tea time is an informal conversation about the talk you've just heard in a relaxed setting. Our question of the week this week is, if you could be any type of crocodile, which one would you be and why? But now it's time for a quick break before tea time. So remember to get up, walk around and have a quick drink of water, come back in 2 minutes. And if you have paleo pets, we always like to meet them. So we'll see you very shortly.
Stay for tea time, I can answer the questions.